There are many types of fascinating microorganisms in the world, many of which have an impact on the health and well-being of humans. We've discussed some of the impact of microorganisms in previous lectures, especially in human defense, pulmonary, and diabetes. This lecture serves to broaden that perspective and reintroduce you to the microscopic world that is found everywhere around us. Many types of microorganisms, or microbes, can impact pathophysiologic processes. We can look at them in categories of effects or biologic categories. We have symbiotic microbes, those which have developed a relationship or mutual single benefit with the human body. We know these as our microbiome, and often this refers to bacteria. They also help communicate, synthesize chemicals, metabolize nutrients, and protect from invasive pathogens. Next, we have opportunistic microbes, which often are neutral or beneficial microbes, which in conditions of immune suppression or immune deficiency can cause pathogenesis. These, once again, are often bacteria, but can be fungi, protozoa, or helminths as well. Our next category are the pathogens, microbes that do not serve a symbiotic or neutral purpose, but are known to cause pathogenesis in humans. These come from all categories of microbes and are significant for their impact on human health. Additionally, we can look at microbes as endogenous, those that are found within the human body normally, and exogenous, those that are introduced to the human body, but are not normally found there. For example, endogenous microbes would be E. coli bacteria in the intestines, whereas Clostridium botulinum, causing botulism, would be exogenous. Additionally, we can view microbes in their biological categories. Bacteria are prokaryotic unicellular and come in a variety of shapes. They are anuclear and do not have membrane-bound organelles in their cytoplasm. Viruses are submicroscopic infectious particles that replicate inside cells of living organisms. A great debate in science is whether viruses are alive or if they are simply complex biological molecular systems. Only around 9,000 types have been explored in detail, compared to the millions of types of viruses known in the environment. Fungi are eukaryotic organisms that have chitinous cell walls and do not photosynthesize, but often are opportunistic decomposers. For pathogenesis, we focus on yeast and molds, not on mushrooms, which are generally not implicated in disease, but may be involved in toxicity reactions. Protozoa are single-celled animal-like organisms that do not photosynthesize and can often cause disease through parasitism in the human body. Helminths, or parasitic worms, are large multicellular animals that perform endoparasitism, or internal action of obtaining nutrients from another organism and doing that organism harm. Intestinal worms, flatworms, flukes, and others can cause disease when they invade and infect the human body. Recently, research has shown a possible link to decreasing allergies, autoimmune disorders, and metabolic syndrome, and the presence of helminths, which is pretty interesting. Microbes are classified by type, size, and reproductive site. For examples are pretty typical diseases that we see when caring for our patients: HIV, hepatitis, chickenpox, wound infections from staph, pneumonia, tuberculosis, athlete's foot, candidiasis, or giardiasis. Okay, so now let's begin examining the process of infection. We know the results of infection are from the immune response: erythema, edema, pain, loss of function, and heat or fever. The process of infection begins way before immune response, though. Initially, the microbe has to make contact with the host. Endogenous pathogens are already found as microbiota on the host, so they have an easy time with encounter. Exogenous pathogens need a route of transmission. This may be through contaminated food or water, and there may be a vector organism, which facilitates encounters, such as another human who has the infection or an animal that carries the microbiome. This is demonstrated by respiratory droplet transmission or transmission through a mosquito bite as an example. The next step is transmission. This can be direct, where exposure to infectious agents via contact with infectious reserves. Vertical direct transmission is exposure from mother to baby via placental transmission, the birthing process, or through breast milk. This includes HIV, rubella, herpes, and many others. Horizontal direct transmission involves contact with infectious blood, body fluids. This includes skin contact, sexual contact, salivary contact via bites, and droplet transmission. Influenza, rabies, West Nile encephalopathy, malaria, and STIs are examples of horizontal transmission. Indirect transmission involves the contact with infectious material, inhalation of airborne pathogens, ingestions of pathogens in water or food, or via zoonosis, contact with animal vectors, typically via animal bites. These include things like malaria, botulism, measles, and food poisoning. In any situation, the encounter with the individual and then the transmission need to occur for the pathogen to begin the process of infection. Colonization is the next step in the process of infection. This is the presence of microbe on a surface with sufficient resources to survive and multiply, but not to cause active infection. In colonization, microbes compete with the microbiome for resources like nutrients and habitable space, while resisting the defense of the immune system. An important concept here that helps differentiate colonization from active infection is that of minimum infective dose, MID. This is the minimum number of microbes required to cause active infection. For example, Vibrio cholera require around 105 microbes to cause active cholera, an infectious diarrhea. Mycobacterium tuberculosis require less than 10 microbes to infect and cause tuberculosis. Another important factor is adherence. This is the process by which microbial receptors bind to the host cells. This is a specialized process and dictates when in the body the infection occurs, due to the receptor and binding interaction. For example, the common cold viruses bind to respiratory epithelium, and Staphylococcus aureus binds to dermal cells, leading commonly to skin and wound infections. Microbes must overcome the initial assault of human defense in order to survive and replicate. They have several strategies to assist them in evading or combating the immune system. Production of toxic substances can kill phagocytic cells, interfere with cytokine signaling, and inhibit the action of digestive lysosomes. Production of antioxidants can neutralize free radicals and reactive oxygen species produced by immune cells. Mimicry of surface receptors in the host allows the microbe to evade activation of complement systems and opsonization, or coating by immunoglobulin complexes, reducing phagocytosis of the microbe. In addition, surface antigen mimicry can lead to type 3 hypersensitivity reactions. This promotes autoimmune reactions where the microbe mimics host antigens and activates immune cells, which then attack host tissues as well as microbes. The microbes can often then change their antigen presentation. And while the immune system attacks host cells, the microbe can survive and replicate. This is a strategy often used by viruses. Mutation of antigens and receptors on microbes can also contribute in the immune system inefficiency, and that rapid changes of antigens and receptors can confuse the immune system and delay responses. Lastly, biofilm formation is an important newer finding in infection. A biofilm is a protective formation of mixed microbes and extracellular matrices of complex polysaccharides that allow the microbes to survive. replicate, and evade the immune system. This forms very often in wound beds of chronic infection and may contribute to the chronicity of the infection. It can also form on implanted medical devices. So once microbes in a biofilm mature and replicate sufficiently, they can break off and form from the formation and become planktonic and then spread to other areas of the body. This is also considered an important process in bacteremia and infectious sepsis formation. Biofilms are especially resistant to antimicrobial substances, antibiotics, and mechanical cleansing. So newer pharmacologic cleansing techniques are being developed that can remedy the presence of resistant biofilms. Our last steps in infection include the invasion or penetration of microbes into tissues. Often this involves evading the barrier defenses and moving through tissue types to find optimal tissues for survival and replication. This is facilitated through natural routes such as blood and lymph vessels, along surfaces of tissues, or through created pathways such as increased vascular permeability, traumatic injury, loss of barrier integrity, and chemical actions that open new paths for invasion. In effect, we can think of the microbes causing damage, triggering tissue death, and then moving deeper into the layers of tissue as a process involved in invasion. Dissemination is the process of microbial spreading, which can involve direct extension, where microbes on one tissue come in contact with another tissue and transfer to that new tissue. Think of bacteria on the skin coming in contact with an eye as you rub it, and then causing infection of the conjunctiva. Additionally, blood and lymph are perfect conduits for spread of microbes through the body, as these natural pathways can move microbes quickly and effectively, if the immune systems can be evaded, of course. Our pathophysiology of infection not only includes inflammation and other immune-mediated responses, but also the destruction of tissues from the evading microbes. Alterations in cellular processes that can lead to damage can be direct or indirect. Direct alterations can be mediated by toxins and is a common process in infection. Endotoxins, which are found inside or as a component of a pathogenic microbe, cause damage to the cell when the microbe is destroyed, releasing the toxin. This is a typical finding in gram-negative bacterial infections such as E. coli or Salmonella species. The death of the bacteria in this case leads to endotoxin release, which then contributes to the pathophysiology of septic shock. Exotoxins are synthesized and secreted by the microbes, and then upon secretion cause host damage. For example, Clostridium species, such as C. botulinum and C. difficile, secrete toxins that lead to adverse effects in their disease processes, like neurotoxicity leading to reduced acetylcholine release in synapses, and C. diff toxins, which damage the cytoskeleton and contribute to cell death and dysfunction in the GI tract. Another common example of exotoxins in the pyrogenic version released by the streptococcal species, which contribute to massive inflammation and cause characteristic bullae, large fluid-filled blisters, and the classic strawberry tongue shown here in scarlet fever, Kawasaki disease, streptococcal pharyngitis, and toxic shock syndrome, all streptococcal diseases. Invasive damage is another type of direct tissue damage. In this case, the microbe causes damage to the host cells via its own growth process. This often involves atrophy of the host tissue through siphoning and use of the nutrients and oxygen by the microbes, leading to ischemia and infarction and death of the tissue. Normally, these invasive microbes would be managed by the immune system, but often the strategies of infection come into play, and phagocytosis is evaded. Additionally, biofilms and protective cyst-like fibrous materials can be formed and help evade the immune system destruction of the microbes. Certain diseases, such as tuberculosis, are known to create protective structures that help evade the immune defenses. Indirect cellular damage can be caused in two ways as well. This involves the response of the immune system and leads to tissue destruction from our own defenses. Immune complexes can form, which consist of multiple antigens binding to antibodies, which then precipitate the antigen out of circulation. These immune complexes are normal methods of managing antigenic invaders. They often get phagocytized and processed to form components for adaptive immunity. In certain situations, though, these immune complexes can end up depositing in human tissues and lead to pathology in those tissues. The immune system recognizes the immune complexes and then reacts to those tissues as if they are the foreign antigens. This is a vast oversimplification of the complexity of autoimmune diseases, but is sufficient for the time being. Listed in the table are typical examples of immune complex-mediated diseases. Notice that rheumatoid arthritis, which we have studied, is included in this list. Cell-mediated indirect tissue damage from infection is caused by the typical inflammatory response. In this case, the normal response to foreign antigens is to trigger the inflammatory processes, which we already know can lead to destruction and dysfunction in the body tissues through phagocytosis, cytokines release, and cytotoxic actions of the immune cells. Often the inflammation associated with infections can cause just as much, if not more, damage than the invading microbe. There are many other influences on the occurrence and pathogenesis of infections. Some terminology to be aware of is listed here. Communicability is the ability of microbes to spread from one host to another. The more communicable a pathogen, the easier it is to spread disease. For example, influenza, norovirus, mumps, and tuberculosis are highly communicable in that they can spread easily from person to person via droplets, the air, or contact. Immunogenicity is the ability of a pathogen to produce immune responses. In many cases, the immunogenicity is good in that the immune system can manage the infection. However, this may also contribute to pathogenesis in that the immune response is so strong, indirect tissue damage is increased, or so weak that the infection can persist unchecked. Infectivity is the ability of a microbe to invade and replicate in the host. This is quite similar to communicability, but involves not just the transfer of the microbe, but the transfer and the ability to cause infection. As we can guess, the mechanism of action of the pathogen also has an influence on the infection. If the pathogen binds to a host cell and causes destruction of the cell, it will lead to more severe symptoms than a pathogen that binds to a cell and leaches nutrients from the cell. Pathogenicity is the ability of a microbe to cause disease and involves the cumulative history of that pathogen. For example, bubonic plague caused by Yersinia pestis is highly pathogenic, as it decimated populations in 3 pandemics in the 8th century, the 1300s, and the late 1890s to early mid-1900s. Since the advent of penicillin, which can kill Yersinia, bubonic plague has become less of a concern, but is still considered highly pathogenic due to the history of the disease. The portal of entry is a concept of how the pathogen enters the host body and can include transmission routes such as foodborne, contact, fecal-oral, bloodborne, airborne, droplet, and many more. An interesting portal of entry is demonstrated by Treponema pallidum bacteria, which causes syphilis. They can easily enter the human body through mucous membranes and through small breaks in skin integrity, which in effect makes skin-to-skin contact a risk for transmission. It was once thought that this bacterium could bore holes through intact skin and invade human body in that route, which has been shown to effect that is not entirely true. Toxigenicity is the ability of a pathogen to produce toxins and the effects those toxins have on pathogenesis of the resulting disease. Virulence is the capacity of a pathogen to cause severe disease. HIV, Pseudomonas bacteria, and Candida auris, a highly damaging yeast, are all highly virulent. Continuing on in our terminology, we need to focus on some epidemiology and the terms used when discussing the prevalence of infectious diseases. We refer to endemic infections, where there are relatively constant rates of disease in a given population. Examples include malaria in parts of Africa and South America, where this disease is commonly found. Another example is chickenpox, common in the United States. These infections occur at relatively constant, predictable rates. Epidemic diseases are infections that have an increase in new infection rates in a population which is larger than the rate typically found. Polio was an epidemic infection in the U.S. in the 19th and early 20th century until vaccinations were initiated. Smallpox was also an epidemic until vaccination eradicated the infection as of 1980. Not all epidemics can be remediated by vaccination, however. In the late 1970s and 1980s, emergence of the human immunodeficiency virus, HIV, was an epidemic in the U.S. and has been reduced to endemic due to advancements in treatment, but is not cured or vaccine mediated at this time. A pandemic, which we should be familiar to all of us, is the epidemic infection, which then spreads to a large area, affecting many populations. COVID-19 is a worldwide pandemic, while in 2009 to 2010, H1N1 or swine flu reached pandemic proportions through the US and the world. Lastly, we are going to examine the stages of infection. This is more terminology for your understanding. We start with incubation, the time frame of initial exposure to the pathogen to the onset of the first symptom. In this time, microbes have contacted the host, entered and evaded, and may undergo colonization, but have not met minimum infective dose. This varies by microbe and may be minutes to hours to even years. Bacillus bacterial food poisoning has an incubation of 30 minutes to 6 hours and is a very short incubation. Kuru disease, a rare neurodegenerative disease found endemically in Papua New Guinea, has an incubation period of 10 to 13 years. Typical incubation periods are 2 to 14 days for many viral and bacterial infections. The prodromal period is the time of initial symptom presentation, which are often mild and include malaise, a feeling of being tired and uncomfortable. In this time, the microbes are replicating and beginning to cause infection. The invasion and acute illness time period occurs when there is a rapid replication of microbes and leads to tissue effects, including tissue damage and death. This is highly inflammatory and leads to more advanced immune system responses. At this time, the signs and symptoms of the infection are more apparent, and often definitive diagnosis can occur. Lastly, convalescence occurs, which is when the infectious agent is removed from the host body and symptoms decline. This is the beginning of recovery from illness, and repairs from damage can be initiated. Alternatively, the infectious agent may not be able to be removed from the host, and damage may be severe enough to cause fatality. Another potential outcome is latency of the microbe, which means the symptoms resolve and the pathogen becomes dormant until later reactivating and causing acute illness. This is best demonstrated by the herpes simplex virus, which infects humans for life and activates in times of stress, leading to cold sores, or varicella zoster virus, which causes chickenpox and can be latent until stress reactivates the virus and causes herpes zoster, or shingles. Latency is highly associated with viral infections, though some other pathogens can exhibit latency as well.